Good evening. Today we're going to finish the section on immunity and immune cells overview, and we will look specifically now at the secondary lymphoid organs, both encapsulated and unencapsulated, and these comprise the lymph node, the spleen, and the mold. What we can see now is one lymph node, and this is one of a huge number of lymph nodes scattered across our body. Each one is about the size of a walnut, and these are all encapsulated. So this is the capsule. Underneath the capsule is the subcapsular sinus, and the subcapsular sinus brings antigens into the middle of the lymph node. As for antigen-presenting cells, dendritic cells, then these reach the lymph node from the tissues via the afferent lymphatic vesicle, and they also empty into the subscapular sinus and make their way into the lymph node. As for the lymphocytes, the B and T cells, these enter through the bloodstream, and they exit the bloodstream through high endothelial venules, and they take this name as a high endothelial venule because these endothelial cells are very tall compared to most endothelial cells. There are two main zones, the B cell and the T cell zone, where lymphocytes accumulate. B cells are here, and T cells are in this region just below the B cell zone. As for the regions where the B cells are, you can see these colored blue. I mean, that's artificial color. These represent secondary follicles with germinal centers. And there are actually two types of follicle, and this is analogous in a number of ways to the situation in the human ovary, where we have primary follicles and secondary follicles that where the oocyte is matured from a primary oocyte into a secondary oocyte. And in a similar fashion, B cells which have entered the lymph node, they've become mature in the bone marrow, they've been selected for their lack of reactivity to self. We call that central tolerance. And these mature B cells, which are expressing IgM receptors on their surface, position themselves in primary follicles. When they are exposed to antigen, then these B cells reorganize themselves within the primary follicle, and they make their way to the periphery of this follicle. And at the periphery, they come into contact with T cells in the T cell area. And T helper cells of the follicular T helper cell class are able then to give them another signal in order to activate them fully. If they become fully activated, then they, along with their follicular T helper cell, actually cause the remodeling of that follicle, and it develops into a much more complex secondary follicle. And this secondary follicle has many zones which are basically responsible for enabling an activated B cell to become the best version of itself, if you like. They enable this B cell to be able to produce very high affinity antibodies, and this is the area where the B cell is undergoing massive proliferation, and where the B cells are differentiating into plasma cells, which are the antibody secreting cells, and also into the memory cells. There's a structure which isn't visible on this diagram, but it's very important for the movement of cells within the lymph node, and this is called the fibroblastic reticular cell conduit, and that's shown up here. And the idea here is really illustrating the importance of non-immune cells, cells which are not derived from hematopoietic stem cells, or stromal cells, if you like. That's stromal cells are cells from the environment which are not part of the main the main cellular structure that we're interested in. So these stromal cells of the type called a fibroblastic reticular cell actually form channels, and these channels enclose long collagen fibers. You may remember from biochemistry that collagen is a trimeric fibrous protein which is very, very long, and it's very important in the extracellular matrix, and it provides scaffolding. The interesting thing about this structure, this physical architecture, is that lymphocytes move along this structure, and dendritic cells actually extend processes through gaps in the fibroblastic reticular cells, forcing there to be a very large surface area for exposure of lymphocyte to antigen. So this system guides T cell movements. T cells actually survive in the paracortex. That's the T cell area, the region below where the germinal centers are, in this area here, for about 16 to 24 hours. And when I say survive, I mean they stay in the area for up to this time. And in that time, as they move along these networks, if they encounter antigen and become activated, then they develop into follicular T helper cells, which, as I mentioned, have their role in activating B cells. If not, then they exit the lymph node and travel to another lymph node. Just to mention that we'll talk more in detail about T cell receptors in the future. We'll talk about the MHC peptide complex, which is presented on the surface of dendritic cells for the T cell receptor to see. T cells can't see native antigens; they have to see them in association with MHC. And we have two major types of T cells: CD4 positive and CD8 positive. The CD4 positives are the T helper cells, and those CD4 positive cells in the lymph node, they become activated to work together with B cells in the B cell zone. These are called T follicular helper cells. So, yeah, I've mentioned that this area beneath the germinal zones is with the T cell zone. But after activation of a B cell and association, forming a synapse, it's called, which with a T cell, the B cell and the T cell actually become a part of the germinal center of a B cell follicle in the B cell zone. And now I want to look inside one of these germinal centers, and that's what we can see on the left here. And there's really a differentiation taking place within this zone, which also has a number of sub-zones. So there's this mantle zone around here, which is full of various types of B cells. A naive B cell, once it becomes activated and becomes a part of a secondary follicle which contains this germinal center, these B cells are actually proliferating in what's called the dark zone. It's called the dark zone simply because under the microscope with certain staining, this zone here appears to be dark. And these cells take the name of centroblast. There are some additional cells that are involved with this, with the development of B cells, called follicular dendritic cells, FDC. And follicular dendritic cells are actually not true dendritic cells in the sense that they are not derived from the bone marrow. It's a it's quite different from a dendritic cell. It has something in common with a dendritic cell, and that is that it is able to present antigen to B cells. However, unlike the presentation by true dendritic cells to T cells, which takes place with the epitopes complex, the epitope peptides complex with MHC, follicular dendritic cells, the name comes from the presence in these follicles, they actually capture free-floating antigens, and and these free-floating antigens are actually immune complexes. So there's something called complement factor, which is part of the humoral system. These are proteins which capture antigen and basically just cross-link it. I say basically, there's a number of things that complement can do, but what we're interested in here in immune complexes is where antigens have been complexed together, and these follicular dendritic cells have a receptor which is able to recognize the body's complement proteins. And if those complement proteins have managed to capture antigens in an immune complex, then it means that the follicular dendritic cell is able to present antigen. So in that sense, it's like a dendritic cell. It's presenting antigen. However, rather than presenting it to T cells on MHC, it's presenting it in the form of captured immune complexes to B cells. In addition, the other aspect of this cell, which is similar to a dendritic cell, is its ability to extend these long finger-like processes through the, and I'll just go back to the last, no, okay, I can't go back to the last slide, sorry. It's able to actually present through these fibroblastic reticular cells, so it's able to extend processes into that area where lymphocytes are passing through that meshwork of collagen fibers within the that reticular network. So that's a dendritic-like property, and that presentation of immune complexes is like dendritic cells. However, no MHC and also no phagocytosis. The true dendritic cell is presenting things on MHC that it has phagocytosed in the tissues and broken up into small pieces and is presenting there. So that's two very important differences to the true dendritic cell. Often the F is written as a small f. So even though it's called a dendritic cell, it's not a true dendritic cell. And this is able to actually activate B cells in the absence of T helper cells. However, the B cells that are activated by antigen on the presence of follicular dendritic cells are not as effective and don't produce a strong secondary response in the same way that B cells that have received T helper assistance can do. Okay, so we've got so just leaving these follicular dendritic cells aside, they play important roles in helping as well to orient and direct cells within this zone, or within these zones. So we have the dark zone and the light zone, and the light zone is divided into two. In the dark zone, these what we now call centroblast cells, which are B cells. These are undergoing rapid proliferation. Once they've undergone proliferation, they then are able to enter the light zone. And in the light zone is where they actually get tested to see which of them has the strongest B cell receptor. One more thing that's happening in the dark zone is that there's a little bit of variation taking place. There are mutations that occurring, somatic mutations that occur in the B cell receptor within these centroblasts here. And that produces a spectrum of B cell receptors that have, some of them will have high affinity and some of them will have lower affinity. Once these B cells enter the light zone, they're called centrocytes, and here they get tested by T helper cells to see which of them has the strongest affinity for the strongest affinity B cell receptor. And the ones that fail the test undergo apoptosis, and they get phagocytosed by macrophages. So this symbol here represents a macrophage. There's a kind of cycling that takes place through this, and it's still subject to people are still studying this cycling process, exactly how it works. But these are the main players: follicular dendritic cells, centroblasts that mature into centrocytes, and they can go through multiple rounds of this mutational process to try and develop even higher affinity B cell receptors. And finally, passage into the this apical portion of the light zone, and then releases either plasma cells or memory cells. Now we come to unencapsulated lymphoid tissue. And this is the mucosal associated lymphoid tissue, also called MALT for short. And the concept here is that the main site of initial infection comes through the skin and the mucosal surfaces. The skin is considered to be the largest organ in our body, and the skin has been postulated to possess a diffuse type of lymphoid organization, and this is called SALT, for skin-associated lymphoid tissue. But this is a controversial subject. MALT in general refers to all the mucosal associated lymphoid tissue, including any skin-associated lymphoid tissue. And what are these mucosal surfaces? The respiratory tract. That's a critical point for infection, and it makes sense that there needs to be something on this front line to be a source of defense, and this is the mucosa-associated lymphoid tissue. In addition, the gastrointestinal tract, a major source of infection from what we ingest, food we eat, as well as the genitourinary tract. So those are three mucosal surfaces which need to have a specialized immune response, and all of them come under this heading of the mucosa-associated lymphoid tissue. When we talk about gut-associated lymphoid tissue, then this is a subcategory of mucosa-associated lymphoid tissue, and this has been called GALT, for gut-associated lymphoid tissues. And that includes the tonsils, the adenoids, the appendix, and the Peyer's patches, which line the small intestine. I will, in the next slide, I will discuss these ones, the Peyer's patches. In addition, there is bronchial associated lymphoid tissue called BALT. This is less organized. Less organized doesn't mean less important. On the contrary, the MALTs are non-encapsulated. This means that the immune system has to be exactly right on the front line rather than sitting behind the skin, waiting for antigens to be drained into it from the lymphatics. So the fact that bronchial associated lymphoid tissue is less organized doesn't mean that this is less significant. On the contrary, this is more significant. This is a huge subject all by itself. The way immune cells are actually circulating around on the mucosal surface of bronchial-associated lymphoid tissues, but that's too complicated for 1 semester 2-hour course. We'll look at the Peyer's patches, which is very well studied as an example of MALT tissue. Um, at the heart of the Peyer's patch is something called an M cell, and these actually are a specialized type of cell which delivers pathogens to lymphoid tissue, which is just underneath the epithelial lining of the gut wall. And this is an M cell surrounded by epithelium in the gut lumen. Now, if we take a cross-sectional view, here we have epithelium, and scattered throughout the epithelium are these individual M cells, and underneath them is this lymphatic tissue, and it has some similarities to the lymph nodes that we saw. There are dendritic cells, and there is a T cell area, and there is a germinal center which sits within a follicle surrounded by B cells, and those B cells which have been activated are within this germinal center and are undergoing the process of proliferating and improving their B cell receptor. So in a way it looks similar. We have efferent lymphatics, so things can travel from here and enter into the remainder of the lymphatic system. But if you notice, there's a very important difference, and that difference is these dendritic cells, they did not migrate here from somewhere else. Rather, these are tissue resident lymphatic cells. They live here, and these are in the lymph tissue. So the story we looked at before was dendritic cells throughout the body and in various different tissues. Once there's infection, then those dendritic cells change their phenotype from phagocytic to migratory, and they travel expressing MHC1 and associated pathogen peptides on their surface. They migrate, they travel to the nearest lymph nodes, and there they present their antigens to T cells. Whereas here, the dendritic cells and the T cells are resident together in this tissue, and this is called a Peyer's patch, and it's something which anatomically can be easily seen at a macroscopic level as a distinct indentation or protrusion on the surface of the intestine at various places. There, I don't remember how many of them, but there are several of these patches situated throughout the lymph node. And these dendritic cells, these little protrusions here can be extremely long. They actually poke out. They manage to push these. They push out dendrites. That's why they're called dendritic cells. They look like nerve cells. If you remember in the study nervous tissue, we have these dendrites. That's how the name came. But they're nothing to do with nervous tissue. So they're actually able to physically poke out a little finger here and pick up and collect antigens which are floating around in the gut lumen, and then they're able to pull those back in and present them. And the M cell has this also unusual property of also being able to trap antigens and process them and present them to the lymphoid tissue here. So this means we can have a very immediate and quick sampling of what's present here, and that can be associated directly and straight away with presentation to T cells. Now, when we talk about T cells themselves also are present within the mucosal surface, and there's a class of cells called from within epithelial lymphocytes, and most of these lymphocytes are actually T cells. So we've got various types of cells lining the intestine. So here are crypts, Paneth cells. There's stem cells here, and they're constantly making new cells which migrate upwards. And that's, you might have studied that, or you might go on to study that in cell biology. You have goblet cells which are secreting things necessary for the mucosa, and we have the standard epithelial cells lining the villi, which are absorbing the nutrients that we want. Not shown, of course, is there's blood vessels here which are taking nutrients to the mesenteric artery to deliver to the liver, and interspersed are these intraepithelial lymphocytes, most of which are T cells. So probably most people perceive T cells and B cells as things circulating in our blood, things which may migrate to tissue at certain times. B cells actually, once they become fully activated and can differentiate into plasma cells, most of them, not all of them, some of them stay within the mantle of the germinal center, but most of them actually go to the bone marrow, and they're constantly pumping out antibodies into the blood. But we actually do have lymphocytes which are tissue resident. And the same is true of skin, but the surface of the skin is highly keratinized. B cells and T cells live, you can find them interspersed within the dermis underneath the epidermal layer, and maybe in the epidermis, the lower parts of the epidermis as well. But here, right on the mucosal surface, we have these intra-epithelial lymphocytes. And it's an amazing number. For every 10 epithelial cells, there's one of these lymphocytes. Now, the last slide I want to show you is the last secondary lymphoid tissue, and that's the spleen. The spleen is encapsulated like the lymph node, but the reason I left that one to the last is because there is another distinction here with the spleen, and that is that the spleen is for surveying what's present in the blood. So here is the spleen within the body, next to the stomach, and here's a cross section through the spleen. Obviously, we have a vasculature which is feeding this, but we can see that there are actually two distinct regions. There's what's called the white pulp and the red pulp, and as we said, it's encapsulated. So we've got white pulp and red pulp. Red pulp is responsible for monitoring and removing any damaged or infected red blood cells, and the white pulp is where basically where the adaptive immune functions are being are developing, and there's a lot of similarity between what's happening in there and the organization and structure of the germinal center within the lymph node. It's not the same identity. It's not absolutely the same. There's not so much polarization in terms of distinct shapes in the different zones of the germinal center, but it is present, or there is considerable similarity between the two. So lymphocytes enter via the blood in both the lymph node and the spleen, but pathogens, they enter the spleen via the blood. So any blood-borne parasites, these will be cleared via the spleen, whereas pathogens that reach the lymph node don't enter via the blood; they enter via the lymphatics. Okay, so that's the end of the first section, and next we will talk about innate immunity. This has been an overview, but in some places it may have been a little bit complicated. The next sections, because they will be more focused by a specific system, I think we'll move more rapidly through those sections. Okay, good night, stay safe, and I look forward to presenting to you again. Any questions? Once I've uploaded this to YouTube, you may ask a question on YouTube, or you have our our WhatsApp group and our e-learning group as well for that communication, whatever is more convenient for you. Okay, good night. Assalamu alaikum.